Good afternoon, ladies and gentlemen. My name is Elias Grampas, and on behalf of the European Parliament Intergroup on Climate Change, Biodiversity and Sustainable Development, I'm delighted to welcome you all to today's webinar, where we'll be discussing more about research and innovation for biodiversity, exploring the potential of gene drive research. So this webinar seeks to provide an overview of how gene drive works and the challenges it also seeks to address, to introduce the most advanced research projects on gene drive in the sector of public health and conservation, and to inter alia present the work that international bodies such as CBD, as well as WHO, are carrying out in this area. Before we kick off, I would like to take the opportunity to thank hosting MEP Ms. Maria da Graça Carvalho, Vice Chair of the Intergroup, for taking the lead with regards to organizing today's event. Also, prior to passing the floor, I would like to invite all participants to send us your questions via the Q&As box of the platform, not the chat box, but please make sure to use the Q&As box. And also, please feel free to indicate the speaker to whom your question is best addressed so that we can facilitate a fruitful discussion to follow. I think that would be all from my side with regards to our housekeeping rules. So once again, thank you very much for joining us today. Ms. Carvalho, you have the floor.
Good afternoon. Thank you very much Elias, for your introduction. Dear speakers, dear colleagues, and everyone connect with us today. I'm very honored to host this webinar, which will allow a debate on a very important topic, yet still rather new and not well understood by the main stakeholders, including by some of the policymakers. I'm referring to the gene drive research. Such awareness raising activities are not just welcome, but also needed when it comes to specific topics like gene editing research, often subject to misconception, misperception, or even confusion. This initiative today is organized in the framework of the European Parliament Intergroup Climate Change, Biodiversity and Sustainable Development. I have been following the debate on genetics-related research for the past decade, even if not always directly involved in the work. First, during my years in the Bureau of European Policy Advisors of the President of the European Commission, especially as a principal advisor in charge of the European Group of Ethics, and it's really called EGE, the European Groups on Ethics in Science and New Technologies. As an independent and multidisciplinary body, EGE advises on aspects of the Commission policies where ethical, societally, and fundamental rights issues intersect with the development of science and new technologies, and on the policies that requires this knowledge of the new fields of science and new technologies. As you might well imagine, concerns in regards to genetic-related research were often raised in the domains such as health policy and agriculture. Then, during my first mandate as member of the European Parliament and as a rapporteur of the specific program in implementing Horizon 2020. More recently, as a member of the Science Advice Mechanisms of the European Commission. It's a service of the Commission that provides independent science advice directly to the College, to the European Commissioners, and inform them on complex issues and that they need to have a decision. Research and innovation and new tools are needed to address conservation and public health challenge. And this debate is timely and relevant one, since the current COVID-19 pandemic has highlighted further challenge related to biodiversity loss and climate change. When it comes to GMOs, the EU is an example to the rest of the world in how it has created and implemented a legal framework that protects its population and their natural resources from possible abuse committed in the name of science. Gene drives, using a variety of genetic engineered tools, hold great potential in the field of infectious diseases, vector and pest control, poverty-related diseases, and outbreak preparedness. Even if there are still scientific uncertainties which need to be addressed and discussed, as well as further investigated. Fortunately, Europe is a center of excellence in this field. Some of the most promising research on gene drive is taking place in Europe, which have built world-class facilities in this research sector. The EU has also supported research in these areas through both the 7th Framework Programme and more recent in Horizon 2020, and in particular for the prevention of devastating infectious diseases, and as the traditional vector control techniques are running up against environmental and resistance barriers. I would like to mention the EDCTP partnership that deals with AIDS, malaria, and tuberculosis. That is a very good example for the world of a partnership of European member states, African countries, co-financed by European Commission, start with 7th Framework Program and has continued in the Horizon 2020. And I was also one of the rapporteurs of the of this of the last report on the DCTP. Compliance with the highest ethical principles as well as relevant national, EU and international legislation is of course a must. When it comes to research fund under the framework programs like Horizon 2020. I believe that ethics must be preserved, even if sometimes it means competing in unequal conditions with more liberal markets. But Europe must also be prepared to revise its legislation and its procedures when there is evidence from scientific research showing that it makes sense to have such a change. Legislation, regulation, procedures, and practice must evolve, also supported by progress of science, and the rational attitude to any fundamental question requires analyzing how urgent is the need to adapt the EU approach. Regular and unbiased debate on promising innovations and technologies is therefore of the utmost importance as a way to ensure that policymakers and legislators, including as members of the European Parliament, are informed and understand the state of play of research, how it's being managed, how it can be used, what are the opportunities will bring, and what are the promises and the risks. 1 year ago, the European Parliament held a debate on the new genetic engineering to CRISPR-Cas9, applying gene driving technology. Early this year in January, the Parliament adopted this position in view of the 2020 UN Biodiversity Conference. This is just to mention some examples of the legislative work carried out at the European Parliament and for which debate, being well prepared and informed, can result in a more productive and content-focused discussion. Having said this, I think that I have shown how important it is for the members of the Parliament to attend debates like the present one. I now leave you with the speech of the specialists, and it is my honor to give the floor to Professor Austin Burt, Professor of Imperial College London, also the school where I did my PhD, and Principal Investigator of the Target Malarium Consortium. Professor, the floor is yours.
Hello. Well, thank you very much for the opportunity to speak to you today. Pleasure to be here. Okay, so I've been asked to give a brief overview of what is gene drive and how does it work. So a few things, what is gene drive? First of all, it's a natural phenomenon of biased inheritance that has evolved many times in many different species. So it was first discovered just as a purely something out in the natural world that people have studied for decades and decades. More recently, it's become a process that scientists are trying to exploit to develop targeted, efficient, and sustainable tools for addressing long-standing questions and challenges, particularly in the control of disease vectors, of invasive species, and of other pests. It is also a bit of a buzzword at the moment, with no precise, widely accepted definition. And so we have things like low threshold drives, high threshold drives, integral drives, tethered drives, split drives, daisy drives, double drives, underdominant drives, sex-limited drives, etc., and so forth, and the list goes on. And this is actually quite a normal thing within science. It's that something becomes a buzzword. It's indicative of it being a very young, active field of research. What engineered gene drives have in common, all those lists on the previous slide have in common, is that they're all LMOs, living modified organisms. So that's using the Cartagena and CBD nomenclature. So there's no legal ambiguity with gene drive organisms, as there is with some gene edited organisms. They all are legally LMOs and hence fall under the current case-by-case assessment regulations. And they all, in addition, in all of the transmission of genes from one generation to the next is in one way or another non-Mendelian. So that's how it works. We get to that. So, and there's a number of different ways that scientists are using to try to get this non-Mendelian or biased inheritance of genes from one generation to the next. And I'll just go through very briefly go through four examples of how that works. In some of the methods that people are doing, trying to address, that when you have a cross between a male and a female, not all the progeny are able to survive, and one or more classes of those progeny fail to develop, and so the bias is created in that way. So, for example, under the cytoplasmic incompatibility approach, if you've got a female without the element and a male with the element, then the progeny fail to develop. And it's the other three classes that manage to develop. The way this thing is organized, that gives an advantage to the plus carrying element, and so that tends to spread through the population. Alternatively, in the Medea sort of gene drive, you've got a different class. You've got eggs either with or without the element, and sperm either with or without the element, and it's the class of progeny that from sperm and eggs that don't carry it, that fail to develop. The other ones do develop fine, and that gives a benefit or transmission advantage in favor of the plus gene, the gene that has the element. Other sorts of gene drive work in different ways. So some researchers are trying to develop a so-called driving Y chromosome, where there's a gene on a Y chromosome that acts to cut up the X chromosome when a male is making its sperm, and so the sperm with the X chromosome fail to develop, and only the sperm carrying the Y chromosome are functional and get all the fertilizations. Or finally, our last example, the so-called homing approach, is when you've got a gene drive on one organism that contrives to copy itself onto the partner chromosome, the homologous chromosome, and so a heterozygous individual is converted into a homozygote. So instead of getting to 50% of the progeny, it gets to 100%. So again, distorting or biasing the inheritance or transmission of genes from one generation to the next. So that's some of the diversity of gene drives under development, and that diversity exists in many different dimensions. So in terms of the molecular mechanisms, gene drives. Some of them are CRISPR-based. CRISPR, of course, is in the news these days because the invention of it was recently awarded the Nobel Prize, but not all others don't use CRISPR. Some of them just involve a single genetic locus. Some of them rely on multiple loci, etc. Many different types of molecular mechanisms. In terms of the intended approach of the gene drive, some of them are meant to suppress the population, the target population, others to replace or change them such that they're no longer harmful. Some gene drives would require to release only tens or hundreds of individuals carrying the gene drive into the population to have the desired effect, whereas others would require releasing 100,000 or more individuals carrying the gene drive to have an effect. After the release, some of them would have very limited propensity to persist in the population after release, and so you would have to have repeated releases in order to have the desired effect. Others, after a single release, could persist for decades or more. And in terms of propensity to spread geographically, some have virtually no tendency in that direction, just similar to the sterile insect technique, which is a method that's been used for decades now to control some pests. And other gene drive approaches have the possibility to spread throughout a species range. So a lot of diversity in many different dimensions. And that diversity of gene drives that are underdeveloped makes a lot of sense because there's a lot of different challenges that can potentially be addressed with these things. And there'd be no reason to think that something for helping control malaria across sub-Saharan Africa would be the same thing that would be something that would be useful for controlling mice on oceanic islands. So we have a large diversity. So there's a diversity of genetic constructs, there's a diversity of target species, and a diversity of receiving environments. And in terms of things that have been thought about over the in the scientific literature, that's literally thousands of possible combinations there. And that diversity of scenarios then, I think, argues very strongly in favor of there being case-by-case assessment of the risks and the benefits associated with each specific proposal. And it also argues strongly in favor of guidance and guidelines being developed that are relatively narrowly focused, that rather than trying to do something that covers all possible cases, which would be very difficult to do, to rather focus more narrowly. So a nice example of that is a paper that came out in 2018 by Stephanie James et al. That developed guidance for quite a specific topic, that is to say low threshold gene drives for malaria transmitting mosquitoes in sub-Saharan Africa. So it's a particular class of construct, particular type of species and receiving environment. And even that took 45 experts and 3 face-to-face meetings in over a year. But at the end of that, they ended up with something very useful, a very useful set of guidance. And so I think that's a much more appropriate level for detailed thinking and consensus building than trying to do everything all at once in a single unified approach. We're not ready for that now. So I think time's up. That's all I have to say. There's just a slide of further reading for those who would like to delve further into this. Thank you very much.
Thank you very much, Dr. Burt, for your very clear explanation of what gene drives are and how they function. Before introducing our next speaker, I would just like to have a follow-up statement on the housekeeping rules. So with reference to participants' questions, when they are related to speakers' interventions, please feel free to forward them to us via the Q&As box, indicating all panelists in the drop-down option that you will have at the right part of your screen, so that we can then, on behalf of the Secretariat of the Intergroup, direct the questions to the most relevant speaker. Moving on with regards to today's agenda, I'd like to turn to Dr. Luke Alfie to better understand some of the purposes that gene drive could be put to. Dr. Alfie works in the emerging field of genetic pest management at the Pirbright Institute, focusing particularly on mosquitoes. Prior to that, Dr. Alfrey was the research director at Oxitec Limited, a spin-out company from Oxford University that he co-founded in 2002. So without further ado, Dr. Alfrey, the floor is yours. Thank you very much.
Okay, thanks, and thanks for inviting me. I've been asked to talk a little about potential applications for gene drives following on from Austin about what they are. So the potential uses of gene drives depend on their particular properties. And a key element of that is that gene drives aim to introduce new heritable traits into target populations. They do that by mating between some released modified gene drive organism, which I will probably now start calling a mosquito because that's what I mostly work on, to go out and seek mates and mate with the wild population. And the trait then gets into the target population through that mating. And that makes these sorts of genetic interactions, genetic control measures, including gene drives, exquisitely species specific, because the modified organisms that you release will mate only with their own species. And certainly for mosquitoes and most animals, that's a very precise thing. Furthermore, they will go out and look for mates for you. And in both cases, they are more specific, and that target seeking ability you won't find from conventional methods such as insecticides. On the other hand, that species specificity means that only the target species will be affected. And if you were a farmer, the crop was being attacked by a dozen different pest species, you might prefer something a bit more broad spectrum than species by species interventions. But a major advantage of such genetic methods is they can access hard to reach and hard to find populations. So if we could through gene drive insert a heritable trait into a target population, what sort of traits might we want to insert? Or outcomes might we hope for? Well, one would be population suppression, so reduce the target number of the number of the target population, which is, if you think about it, what most current pest control aim to do, what chemical insecticides and so on aim to do. And one could imagine putting a sterility trait or something like that into the target population so that enough of the pests gain this trait and the target population will decline because of that sterility that you've driven into the population. A different approach, aiming is to modify the population to try to reduce the harm that it causes without particularly reducing its number. So this is really relevant only where the pests are doing some sort of indirect damage, so perhaps not directly feeding on a crop or on a target, but transmitting disease like mosquitoes do. So, mosquitoes biting you is annoying, but it's really the malaria or dengue or whatever disease transmission one is trying to stop. And so one might imagine spreading resistance to dengue or malaria through a mosquito population. So the mosquitoes are still there in rather similar numbers and not obviously different, except that they can't transmit the pathogen. And this outcome, if you think about it, is conceptually very similar to vaccinating the pest population, which of course we can't do with mosquitoes for any number of reasons. But if we go out there and vaccinate all the mosquitoes, you would have the same outcome. You'd have a mosquito population living in the area, looked pretty similar to the way it did before, in fact, indistinguishable really from the way it did before, but no longer transmitting the disease, and so not doing the harm that we are largely trying to avoid. A key feature of gene drive is the single target species I've mentioned, but another aspect is, again, because it's mating and inheritance based, time for these applications is in generation time. So if the gene drive can increase in frequency per generation, what that means in calendar time depends on the generation time. And so applications have overwhelmingly focused on relatively short generation species, so things with generation times of weeks or months. so that you get quite a few generations in a year, and you see things happening over a time scale of a small number of years, rather than the sort of decades or more, the longer generation in time organisms, so things that had years to generation might take. In terms of population suppression, rodents, mice, and rats on islands for conservation purposes. Mosquitoes, so most of the research so far has focused on mosquitoes. And so that might be malaria, particularly in sub-Saharan Africa, and dengue and other mosquito-borne viruses, which are transmitted by different species and somewhat in different places, as public health applications. And then but also mosquitoes are relevant for conservation as well. So avian malaria, for example, in the Hawaiian Islands is a major problem for some unique bird species. There are a few examples with agricultural pests. Spotted winged Drosophila, a pest of soft fruit, is one of rather few examples that I'm aware of where people are at least thinking about the possibility of gene drive approaches. Then for population modification, so this is, remember, trying to modify the population to be less able to transmit disease, but it's really disease transmitting organisms that are relevant. So again, mosquitoes, for both for malaria and for other viruses, dengue and the other viruses that other mosquitoes transmit. You'll see that all of these target wild pest populations of one sort or another. And I'm not aware of obvious applications for domestic species, so crop plants and livestock and that kind of thing. And the reason really is that their breeding and mating is already under human control. And so artificial selection for desired traits, very powerful tool for spreading those traits into populations where we control their mating without really any need for gene drive. So I see few or no obvious applications for gene drive in those areas. And the current developers are overwhelmingly universities and public research institutes funded by governments and philanthropies, and I don't really see that changing much in the near future. A couple of examples. Malaria in sub-Saharan Africa. The malaria causes about 500,000 deaths a year, mostly in children under 5 in sub-Saharan Africa. This is actually getting a little better. This is down from 1 million plus some years ago, and this is through the use of bed nets, enhanced spraying, and so on. But those methods, though effective in reducing the fatality rate in cases, have not proved adequate to get much below that. And so complementary new methods, complementary additional methods are very much desired. And genetic methods and gene drive have a lot of potential in that area. So, of course, malaria is a mosquito-borne disease. Human malaria is exclusively transmitted by Anopheles mosquitoes. And the most important species is Anopheles gambiae in sub-Saharan Africa. So in terms of gene drive, what might we do? Well, spread a reduced fertility gene or in the same vein, a sex ratio distorting gene, so essentially makes them all male, through the target population or species. And target malaria that Austin Burt leads is an example of that, with more information at that web address. And the aim there would be to reduce the number of relevant mosquitoes below a threshold needed to sustain malaria transmission. And an alternative approach, for example, from the University of California Malaria Initiative, would be to spread genes into that mosquito population that makes them unable to transmit the malaria parasite. And there, the outcome would be the mosquito population would look largely unchanged, but it would be unable to spread malaria. And if you could get that malaria blocking gene into enough of the mosquitoes, again, there wouldn't be enough mosquitoes that can transmit malaria to sustain malaria. And in both cases, the aim is to spread this modification broadly through or throughout target species, for example, Anopheles gambiae. The second rather different example, rodents on islands. So islands are 5% or so of terrestrial land area, but they're often evolutionarily distinctive because of their isolation and very vulnerable to threats from outside. So introduced species, introduced disease from outside, where they have evolved in the absence of those predators or diseases. And so a large majority of vertebrate land extinctions have occurred from on islands, and invasive species are implicated in the large majority of these. with rats and mice being particularly important. I'm sure you know they eat eggs and attack birds, particularly birds that can't fly and so on. There are successful examples of removal of rodents from small islands, but with current methods, which is basically mass poisoning with warfarin derivatives. But it's very hard to do, and the methods are, well, the mass poisoning approach is not ideal, and not just complementary, but actually replacement methods would be highly desirable. Gene drives may be able to do this. So again, if we could spread a reduced fertility or sex ratio distorting gene through the pest population on the island, with an aim to eliminate it from the island. Now, in this case, the aim would be to spread the modification through the island population only, not substantially onto island populations, and certainly not to invade and spread, on mainland populations, certainly not to invade and spread through those mainland populations. So that would be looking for a different sort of gene drive that would stay in the target system, target population, but stay in the target population in which it was introduced. And as Austin said, there are a wide range of gene drive designs that are capable of greater or lesser degrees of spread and confinement and so on for these different applications. I would say that the development of gene drives in rodents is much less developed than it is for mosquitoes, but we know a lot about the genetics of mice from their long use in laboratories for a whole range of other purposes. So the basic genetic knowledge is really very good for developing these sorts of approaches. So just in summary, there's a wide range of possibilities I illustrated too, with rather different purposes and target species, and also characteristics in terms of desirability of spread and persistence and so on. But there are many more. The targets will generally be these single major species with short generation times, and typically wild pest species for the sort of reasons that I mentioned earlier. That's it. Thank you.
Thank you very much, Dr. Alfie, for stating plainly the potential advances that could be made using gene drive-based technologies to address a number of crucial challenges in both conservation but also public health. Bearing that in mind, I think it's crucial to now turn to a discussion on aspects of research related to governance, oversight, and ethics. So a pleasure to welcome Ms. Camila Bich. Camila is an independent regulatory expert specializing in novel technology products where regulatory requirements are uncertain or require development in both science and policy areas. In addition, Camila has over 20 years of international experience in the regulation of a wide range of novel biotechnology products, while for the last 10 years, she has specialized in pioneering and navigating pathways for the regulation of genetically engineered insects. So without further ado, Ms. Bich, the floor is yours. Thank you very much.
Thank you. Thank you very much to the European Parliamentary Intergroup for inviting me today. Can you see the presentation yet?
We can hear you very well, but we cannot see the presentation.
Okay.
I see it's popping up now.
I think it's popping up now.
Okay.
All right, thank you very much. Okay. Okay. So today I've been asked to look briefly at the regulatory frameworks for gene drive organisms. I have to say it is very brief because there is a lot out there. So happy to take any questions later. The first thing I'd like to say is that gene drives, as Austin has previously said, are still at a very early evaluation stage. In 2018, a CRISPR-Cas9 gene drive was published that had suppressed a small cage population in Anopheles gambiae. And this is still in research, and it's still at a very early stage in looking at larger cage populations now. And I think it's really important to say at this point that no gene drive has entered field trial stages. and no regulatory authority anywhere in the world has reviewed an application for field release of gene drive as far as we're aware. So it's still very early stages. However, taking advantage of that early stage evaluation, that many organizations have been publishing guidance for both risk assessment and policy on gene drives. These include the National Academy of Sciences in the US, who published in 2016, but also the European Academy's Science Advisory Council, publishing on genome editing, which also include gene drives at that point. The African Union, where many of these technologies related to mosquitoes may be introduced, has also published in policy areas and recommended the continuing use of gene drives as new tools for malaria control. And they're currently preparing regional guidance for West Africa on how these tools may be integrated with other vector control mechanisms for malaria. National authorities have also published documents on gene drives, two particularly from Europe that are quite prominent, the High Council for Biotechnology in France, and also the Dutch COGEM report, I believe, which will be discussed further later on today by Patrick Riddleshine. There are some common themes emerging from these documents, especially the recommendations for risk assessment, that risk assessment for gene drive should incorporate socioeconomic impact assessment. It should use ecological quantitative risk assessment and mathematical models of scenarios to assist with planning release of gene drive. It should also learn from the existing biocontrol and vector control programs that are seeking to control mosquitoes or agricultural pests, and include benefits to have a comprehensive risk-benefit assessment. And today I'm going to talk specifically just about two documents, one from the Convention on Biological Diversity and one from EFSA. So the Convention on Biological Diversity ad hoc technical group on risk assessment document was published in April 2020. They already had guidance in the Protocol on Biosafety, Annex 3 risk assessment methodologies, which are suitable for gene drive, but they wanted to look whether additional guidance was needed for gene drives specifically. So this RTEG was convened to examine that. Their recommendations from the report published in April 2020 recognise that risk should be balanced with benefit in decision making, that gene drives are LMOs and fall within the scope of the Cartagena Protocol on Biosafety, that existing risk assessment frameworks may be applicable, although additional attention is required in some technical areas. The analysis should be case by case and a thorough risk assessment conducted prior to release. And public consultation, including Indigenous peoples and regional cooperation, should be included and considered. The RTEG ultimately recommended that preparation of additional guidance on these specific technical issues was warranted, and a further discussion will be had at the COP-MOP 15 on preparation of additional guidance materials. Turning now more to EFSA and to the European activities. EFSA got a mandate from the European Commission in June 2018 to identify potential risks that gene drive modified organisms could pose. They were asked to identify novel hazards and what appropriate comparators might be to compare against, and also to determine whether existing risk assessment guidance documents were sufficient and where updates might be required. EFSA conducted 2 stakeholder engagement evidence sessions, one in May 2019, a workshop, and a further meeting in October 2019. They've published their draft opinion for public consultation in April 2020. And some of the conclusions included that environmental risk assessment for gene drive can be built on the existing risk assessment framework, that a case-by-case approach is necessary, and that should be based on systematic problem formulation methods framed by the relevant protection goals outlined in law and experiences with other pest or vector control activities. They also recommended that the guidelines should be updated in specific areas, including the use of mathematical models, molecular characterization, assessment of persistence and invasiveness, and post-market monitoring, or post-release monitoring, probably in the case of gene drives. Further information, that document's available and available at this website. The final opinion is expected to be published later this year, sort of November, December time. So that'll be interesting to see that opinion. I also want to mention today, in some countries, there is a legal requirement beyond biosafety or environmental risk assessment, and that's to conduct an environmental impact assessment. The environmental impact assessments not only evaluate the impact to the environment, but also to health and potential and socioeconomic impacts. They also look at cumulative effects and transboundary effects, and include structured public participation and feedback. There is well-established guidance on how to conduct an environmental impact assessment, and this is from organizations such as World Bank and International Finance Corporation, and they're widely used, particularly for development projects. The outcomes identify both positive and negative impacts. They identify information gaps. They include management options. They look at the position with alternative technologies of the new technology. And they also include follow-up audits. So I'd like to summarize in my last few seconds that the regulatory approach for gene drives can build on existing regulatory frameworks, GMOs, and biocontrol solutions. That case-by-case evaluation is being recommended in many of the guidance documents, especially used with a transparent and scientifically rigorous risk assessment methodology. Additional guidance is needed in some areas, such as the use of mathematical models, persistence in the environment, post-release monitoring. But when we're talking about gene drives, we should include benefits, socioeconomic impacts, and stakeholder engagement in the decision-making process. And there is an increasing number of guidance documents available to guide the research as it currently is happening. So we're not operating in a regulatory vacuum. So thank you very much.
Thank you very much, Camila, for giving us a clear picture on how regulators and policymakers currently assess the risks, benefits, and potential impacts of this technology. Before I provide the floor to Dr. Emerson, seeing that we're running just a couple of minutes late, I would like to thank our previous speakers for their kind professionalism, but also take the opportunity to remind our speakers to follow to please respect the allocated time given for their interventions. Moving ahead, I would like to turn to Dr. Claudia Emerson to present how ethical concerns regarding this research are being addressed. So Dr. Emerson is the founding director of the Institute on Ethics, Policy for Innovation at McMaster University and associate professor in the Department of Philosophy and also associate member in the Department of Medicine, Faculty of Health Sciences. Her work in applied ethics considers ethical issues and policy gaps in global health research, while she has been also working with various stakeholders in the field for more than a decade, examining issues that arise along the discovery to delivery pathway for potential life-saving interventions. So Dr. Emerson, it's a pleasure having you with us today. The floor is yours.
Thank you very much, Elias, and thank you very much for this opportunity to speak here today. I'm delighted to be able to participate in today's conversation. So, and the screen is visible now, I assume. Okay. So thanks again for the opportunity to speak on this important and timely topic. For me in particular, it's of tremendous interest as someone who looks at ethics and policy issues in global health research. And in particular, I'm interested in the applications of gene drive technology for public health, much along the lines that Professor Burt described. And one of the things that I was thinking about in preparing for today's talk was that in our limited time, we are probably unable to go through a consideration in any depth of the kinds of ethical issues that impact gene drive. And so what I'd like to do today is actually highlight 3 points that I think are ethically significant and I think very important for policymakers and decision makers to think about when they consider the role of gene drive technology in addressing potential challenges that we're facing in public health, in conservation biology, and in agriculture. And so my remarks today are then framed from this perspective of public health. And so the first point I'd like to make is that while the technology is new, as Professor Burt highlighted, the ethics issues are not. And there tends to be a tendency, a common perception that when a new technology comes onto the scene, when something new is emerging, that the moral issues that we have to grapple with and that we have to address are similarly novel and new, and we are unprepared, sometimes ill-prepared, to deal with them. And the unfortunate consequence of that misconception is that it seems to amplify or compound the perception of uncertainty or risk that is associated with the technology. And the reality is that the ethics issues are in fact not new. So the kinds of issues that we have to grapple with around gene drive technology, and in fact any emerging technology, are ones that we have encountered in the past. They are ones that we face now, even in a context of more familiar technology. So questions around how to assess and balance risks and benefits. How should we engage the public? How should we determine the nature and scope of informed consent and whether consent is fair? And questions of accountability, transparency, and governance and oversight, as Camila, my colleague, highlighted. And so these are the sorts of questions that we ask about all sorts of science, technology, and innovation. And in fact, we have plenty of guidance and have standards and norms at the international level to govern research with human subjects, animal welfare, risk assessments, and we even have specific guidance around genetically modified mosquitoes for gene drive, specifically for public health applications. In 2014, the WHO issued its guidance, and there is an updated guidance document that is presently under preparation, and my understanding will be released in 2021. And so, and as Camila highlighted, there is other guidance and regulatory frameworks. And so importantly, we have then some guidance and frameworks to look towards. And perhaps we need to adapt, perhaps refine in some context, given the case-by-case approach that is being considered for gene drive technology. Nevertheless, there are tools and there are guidance documents there. And there is even a community of practice that has, that has emerged within this field with funders as early as 2017 preparing and committing to principles for gene drive research that set very high scientific and ethical standards for which they said that no funding from the organizations that subscribe to these principles would be issued unless these principles were taken up. And so questions of accountability, transparency, safety are paramount in this work. And similar work is underway in the science community among scientists who want to subscribe to a code of conduct. And so the ethics issues are not new, and we in fact have ways of considering them and addressing them. The second point I want to highlight follows from the fact that we do have these frameworks and we do have guidance to look towards. And that's related to how we then understand and how we assess the issues. And in particular, I want to highlight as an example, risk-benefit evaluations. And you noted in Camila's presentation on the slide, she had a point around that the potential benefits need to be considered along the risk benefits. And the reason that is that we do a very good job of conceptualizing and thinking through and parsing out the risks related to potential new technology, and as we should, because risks are ultimately the signposts that signal the kinds of harms that we're trying to avoid. But we do a comparatively poor job of highlighting and parsing out and getting towards the benefits. And this is problematic primarily for two reasons. In the first instance, it doesn't give us a fair assessment. So a comprehensive accounting of the risks and benefits is essential if we want to do fair evaluations of the technology. And the second point is that it obscures other commitments that we have in relation to the broader sustainability and development agenda, to which these types of technologies might play an important role. And so as an example, when we think about the benefits, for example, of gene drive technology for a public health application, we often talk about this as, well, this has the potential to eliminate a disease such as malaria, which is a tremendous benefit indeed. And we might even say, and this will confer tremendous socioeconomic benefits, and it might even get into the numbers of, would save hundreds of thousands of lives every year, would avert millions of infections, but we don't go down far enough. And so if we start to unpack that kind of benefit, the elimination of disease, we start to think about things such as who is most adversely affected by vector-borne diseases. Well, they disproportionately affect people who live in low and middle income countries, who may often face conditions of poverty, background conditions of social injustice, and have diminished opportunities to healthcare and education. If we unpack that even further, we get down to a level, for example, in the case of malaria, among those people, who then is then much more affected? Well, children who are particularly vulnerable, and then women, caregivers, mothers, grandmothers, ones who have to care for sick children and dying children. And so they also then face diminished opportunities for gainful employment outside of the home, access to education, and whatnot. And so then when we're thinking back to, well, the public health benefit of eliminating a disease such as a malaria with a powerful technology actually now has this very gendered dimension. And so getting very comprehensive and very precise about these types of what the risks entail, but also what the benefits entail, help to remind us of the kinds of commitments that we've made in terms of the global agenda around sustainability and development, and highlight these more obscure features around gender equality, around equity, and it's important to think about those because we don't have all of the tools that we need in order to make good on these commitments and to follow through on obligations. So it's important to think about what role something like gene drive technology might play in these types of considerations. And so the last point then I just want to highlight is that the justification for moving forward with any kind of research, particularly research with risks, is that it has scientific and social value. Ultimately, the scientific and social value of any kind of research endeavor must outweigh any sorts of risks. And so in trying to get towards the scientific value, that's probably not very difficult to do in terms of technologies that have good potential, because science gives us facts, gives us data and evidence, and facts are supposed to be indisputable, not really open to debate. But values, on the other hand, in a pluralistic society with a multiplicity of values are often incommensurable, and there is no way to resolve fundamental conflicts between deeply held philosophical beliefs and the multiplicity of values. And so how do we approach something like this? How do we then get at a full understanding of what the scientific and social value might be for something like gene drive technology? Well, we have to kind of pull back and take a global lens to this matter. And our current situation in the pandemic actually is an important lesson in reinforcing things that we already know about climate change and environmental degradation, that problems at scale and that global problems demand global solutions and demand a measure of solidarity, which has emerged as a key value in the fight against pandemic. So when we think about what a gene drive solution might be for, say, vector control to eliminate malaria, it would obviously hold tremendous social value in a place where disease is endemic, perhaps little or no value where that is not a concern. But that isn't the way we should be looking at it, at least from an ethics perspective. It shouldn't really matter that there isn't malaria in Portugal, the country of my birth, or in Canada, the country where I live. It only matters that there is malaria still in the country, in Africa and in other countries. And so we need to think about these broader values that have universal appeal. And we need to think about the broader social value that comes from addressing public health problems, and ultimately, even if the diseases don't spread, their negative impacts do. And I will stop there and say thank you very much for your attention today.
Thank you very much, Dr. Emerson, for giving us an overview of the ethical issues around gene drive, putting in evidence that despite the novelty of the technology, many ethical issues remain similar to methods and strategies used in the past. Moving ahead with our agenda for today, I would like to turn to Dr. Patrick Rudelsheim to inform us about regulatory developments and decision-making processes related to gene drive. So Dr. Rudelsheim is a general partner of Perseus BVBA, a service company focused on biosafety and related regulatory development requirements. In his capacity, Dr. Rudelsheim offers his extensive and global experience in every aspect of biosafety and biotechnology regulation to a range of users and developments. So Dr. Rudelsheim, thank you very much for accepting our invitation to address today's topic.
Thank you very much to speak at this forum. I hope everything is connected and you can hear me well. I was asked to specifically address also again regulatory developments and decision making, and we are actually a service focused on providing services for biotechnology, in particular biorisk management and complying with the biotech legislation. So we're really into the practical part of the regulatory side. What I wanted to share with you, and actually presented is the outcome of a research project that we have done for the Dutch Advisory Committee on Genetic Modification. That's an independent advisory body to the government. They advise and they inform. And in 2018, they've issued a request for a research project on gene drives in the environment, looking at the experience in order to inform the risk assessment. So what came out? First of all, again, following up on the other findings, is the fact that indeed there have been natural and synthetic gene drives are being explored. As was pointed out, for the synthetic gene drives, that's still relatively new and they're not very advanced yet to the stage of fields. But for some of the natural gene drives, there have been applications. There have been some experiments, we can learn from that. But again, if you look at the natural gene drives that have been used, then in fact there is one system which is already quite advanced. And although it was not based on GM, it has been also subject to risk assessment. And clearly we can learn from that. Out of all of these things, given the fact of the diversity, and I take the point of previous speakers, given the fact of diversity of the different gene drives, of the environments, of the organisms in which they are used, clearly a case-by-case risk assessment remains very important. To really cut it very short, I think there's much more, of course, in the document, but we have not seen any indication so far, although of the limited experience, but we've not seen any negative indication on human health or the environment. Yeah. Clearly, again, part of the risk assessment deals with addressing all the possible concerns. So some people may say it's not awkward that you don't find anything because everything has already been taken care of. But it also shows that these kind of approaches can work and live up to the expectation. What is important is that although we may talk about gene drives as having a tremendous impact, they remain very delicate constructs. Scientists will tell you how difficult it is to design it, but it's not something which is created by accident, but also even if you have them in place, they are subject to many influences. And therefore, the impact may actually be much, much lower and much more controlled than what we originally were expecting. An important one in relation to this is then to say that the concern over the release of gene drives, where people may have worst case scenarios of entire species being wiped out and so on, may actually not be as dramatic and should definitely be nuanced. It should be further put in context to understand really what the impact is. I realize it's in a nutshell, also in a matter of keeping up with time. There's more to read in the document. But from that document, from that study, actually the advisory committee then had further debates and has issued a proposal to the government, to the Dutch government, which some of the points that they include are indeed the fact that there are no indications today that these gene drives are really uncontrollable. Of course, it depends on the gene drive and the application, but it's not something which would be a general concern. They also indicate that it's not necessary to include all the CRISPR gene drives automatically in the highest containment level, that you really have to look at case by case, and that you can determine these measures based on the standard procedures. So this is the deliberation of the committee itself. We actually provided the background information, but clearly the committee also took other aspects and their own expertise into consideration of that. Now, out of that, as I was already indicated, we have EFSA, the matter in more detail, also in relation to their own guidelines. This has been one of the inputs that has been provided during the EFSA process. Camilla was also referring to the work of the ATIC of the CBD. That indication or those indications were also provided, asked to base or to use our experience to make the first information package for this process of the ATIC. Important, though, is that whereas the COGEM was looking at the broad range of systems, including the natural systems and the synthetic ones, both EFSA as well as in particular the CBD was focusing specifically on those gene drives synthetic. And clearly, if you really want to build on experience, then it's likely that you would not take into account on one hand the experience, but also the potential impact of natural gene drive systems, because in essence, whether it's synthetic or natural, the impact may be very similar. Already coming to some of the key points that I want to bring in this presentation, again, reminding that there is some overlap with previous speakers, but clearly the diversity of gene drives must be considered, and it must be considered in a broader picture. Not only the diversity of the gene drive systems, also the type of applications and the alternatives. And I think it has been pointed out that even today, of course, there is already vector control happening. Sometimes with practices that we would prefer to reduce. Well, gene drives may offer an opportunity there. They come with their own questions, but we should look at that in the broader picture. Second point indeed has to see that the regulatory setting, which is based on the precautionary approach and which requires risk assessment, is preparing for this. The COGEM, when they looked at this in 2018, they were not immediately considering an imminent release in the Netherlands, but they were already anticipating and preparing and evaluating, just like EFSA is evaluating the guidance. I think it's clear that the regulatory setting is there and that they're preparing ahead of the introduction of new systems. Within that precautionary approach, a gap in uncertainty analysis is one of the key elements. So understanding where the gaps are, where are the questions that we still want to see further addressed, is important. And I think we still have a discussion ongoing at this point, where we say, like, what is really the novelty? What would be the specific feature of a gene drive that we still need to look at now before we can move forward? Because many of the points that we see are indeed in line with what we already have looked at for other GMO. And finally, I like to point out from the experience that we had in going through these different evaluations, is that even before there is the first release, it's probably one of the technologies where people have really been looking very carefully on what could be the potential risks. Not only what could be the potential risk, but also can we identify systems that can help us to cope with that even ahead of us producing those risks. I think that is an important one which completely fits within the precautionary approach, but which in many cases is maybe not highlighted enough. And it was just showing some of the approaches that are used, where you say there's different levels of containment, even within the science itself in terms of the molecular or the genetic structures. So those are important elements that have been taken up, and probably in a way that we haven't seen for any other technological development. And for the sake of time, I think I would like to keep it to this. Thank you again.
Thank you very much, Dr. Rudelsheim, for providing us with further examples of practical guidelines and also regulatory requirements for gene drive research. And for the last but not least part of this webinar, I would like to turn to experts who will present the work of the International Union for the Conservation of Nature, IUCN, but also WHO, the World Health Organization, related to gene drive research. To begin with, I would just like to provide the floor to Dr. Kent Redford, who serves as chair of IUCN's Task Force on Synthetic Biology and Biodiversity Conservation, which published last year a very interesting and highly relevant assessment of synthetic biology and biodiversity conservation, entitled Genetic Frontiers for Conservation. Mr. Redford, it's a great pleasure to have you with us today. So without further ado, you have the floor. Thank you very much.
Thank you very much. It's my pleasure, but to give a presentation as chair of the IUCN Task Force on Synthetic Biology and Biodiversity Conservation. And we very much appreciate the opportunity given by Dr. Maria G. Graça Carvalho and the EU's Intergroup on Climate Change, Biodiversity, and Sustainable Development to be in front of you today. So I'd like to start out by asking you who are watching this to think about cows. And when we think about cows, we often think about the products that come from cows: leather, meat, and milk. What if we could get these cow products without the cows? In fact, that is currently possible, at least in research stage, because products from cow genes have been inserted into algae using the tools of synthetic biology. And these companies, New Harvest, Perfect Day, and Modern Meadow, are preparing to release synthetic, though cow-based versions of meat, milk, and leather. These tools of synthetic biology come out of this field for which there is not a universally agreed upon definition. And so we offer here the one from the Convention on Biological Diversity. Many of you will be familiar with this. Basically, further development of modern biotechnology. It accelerates the understanding, design, and redesign of genetic materials. And synthetic biology is most advanced in the fields of agriculture and human medicine. In agriculture, there have been a wide variety of applications, including the attempts to transform the carbon photosynthesis in less efficient plants to become more efficient, increase micronutrients, create new crops, increase drought tolerance, disease resistance, nitrogen fixation, and then a variety of uses proposed and implemented in animal production. And then in human medicine, the other of the two areas where this is most advanced, including much of the conversation we've heard today so far about reducing malaria burden, also developing new and improving drugs, drug effectiveness, monitoring diseases, and improved nutrition, amongst many others. Much less developed, though in the early stages, are the applications of synthetic biology and its tools to conservation. And in the assessment that we did, we looked at these 8 different proposed, and I stress proposed, applications. Already covered is the rodents, invasive rodents on islands. Also covered by Luke was the invasive mosquitoes for bird conservation in Hawaii. We also have ideas on how to, using these tools to conserve black-footed ferrets, to try to reintroduce the American chestnut, to equip corals to be better able to respond to ocean changes, to replace the use of horseshoe crab blood, to use it, as we've discussed, but many others have discussed, about vector suppression for malaria, and then to look at honeybee colony collapse. So the IUCN was requested by its members at the World Conservation Congress in 2016, in this Resolution 86, to develop an assessment. And what I'm going to be speaking about today is the results of that assessment process. And I want to stress that we were asked to look at the wide variety of synthetic biology tools, and GeneDrive was included as one of these tools. The task force itself has a broad range of members: natural and social sciences, governments, NGOs, academia, business, indigenous folks, and virtually all parts of the world represented. This task force then created a technical subgroup who were the lead authors of this assessment that I am summarizing today, and it had the same kind of diversity as the task force itself did. So in May of 2019, we published this, which is available on the web through IUCN in English, Spanish, and French. And so it's entitled "The Genetic Frontiers for Conservation: An Assessment of Synthetic Biology and Biodiversity Conservation." We released a draft of this to a broad number of stakeholders. We sent out over 10,000 invitations to review. We got over 500 comments back. And one of the important things that came back in this initial review was that we needed to be better balanced in discussing both the potential negative and potential positive implications of the conservation applications. And so we rewrote the document. We're working on trying to achieve much more of this sort of a balance. The assessment looks at 2 streams of ways in which synthetic biology can affect conservation. On the left-hand side, and those are the kinds of things that have been reviewed and that we think about, are those that reduce threats and increase resistance. Those are the direct ways. But perhaps more important, certainly equally important, is the implications of applications that are not intended for conservation benefits, but might have conservation implications. These include things like pest control, product replacement, agriculture, of course, and environmental engineering. So these two streams then unite in their implications for their effects on genes, species, and ecosystems, the three important components of biodiversity. So the assessment is a long one, and it's very heavily literature-based. These are the key messages that I'm hoping to cover here with you. Just very briefly, the first is that there are conservation implications, important implications that need to be looked at because the current set of tools are really not allowing us to achieve our desired global biodiversity goals. Second is that conservation really needs new tools. The existing tools by themselves are not sufficient. The third is that synthetic biology is growing very rapidly. Its major applications are not in conservation, but in agriculture and human medicine, and they're moving at a very rapid pace. Fourth, that engineered gene drive, the major focus of this session, is unproven, but it has great potential, both positive and negative in a conservation context. Fifth, that there are potential conservation beneficial impacts, possible but still untested. Also possible but untested are the detrimental conservation impacts which might take place. Seventh is that the discussion that we had very broadly about this topic has revealed the fact that values and worldviews very heavily influence the development, assessment, and in fact, the ways in which people receive the news and the discussion of the possibilities of this field. 8, that indigenous and local communities must be key actors in the development of research and decisions. 9, that the committee believes that the existing structures of governance may be challenged, but may be robust enough to handle these governance questions. And 10th, that the assessment itself is not a risk assessment, but rather it's an assessment of the potential across a broad range of fields. All of these conclusions in the final assessments are rated against these qualitative uncertainty terms. So you would be able to see that we looked at the level of agreement and consensus and the quantity and quality of evidence in presenting these conclusions. So where are we with this? The assessment was published and it led to a draft set of principles which were part of a motion that was going to be voted on by the IUCN membership. And these principles include the following: that the biodiversity conservation imperative is strong, that we must look at intergenerational equity and sustainable development. We must have respect for rights, beliefs, and cultures, stakeholder and rights holder participation. Of course, free, prior, and informed consent, an important evidence-based view about these things, that they need to be multidisciplinary, looking at both the conservationist and the synthetic biologist. And of course, that the ethics position on this, as discussed by some of my colleagues, is critical. So we also laid out a set of key considerations for applications, the first of which has been widely mentioned by my colleagues on this call: the importance of case by case, the importance of evaluating existing alternatives, that is, not using these to replace what we're currently doing, but rather to complement them. That we need to go through steps to ensure that they don't threaten biodiversity, that this assessment should be staged, governance a key issue, knowledge gaps and research needs must be filled. There are important parts of transnational knowledge transfer. And then there are discussions about the potential introduction of moratoria that we address in this. So where are we now in the process? We were to have voted on this motion at the World Conservation Congress in June in Marseille. That was postponed till we don't know when, in 2021. So right now we're in a holding position in this regard. And finally, just a way of reflecting on the current state we're all in with the COVID-19 pandemic, it is our belief that it may be that the ways in which synthetic biology tools may or may not be used to develop vaccines for this pandemic may influence the way that the larger public will view the potential applications of these tools to biodiversity conservation. Thank you very much.
Thank you very much, Mr. Redford. for so clearly laying out the significant work undertaken by IUC on this. Now I'd like to turn to Dr. John Reeder, who is Director of TDR, the Special Program for Research and Training in Tropical Diseases since February 2012. Moreover, Dr. Reeder is the Director of the Research for Health Department at WHO in Geneva since 2019. So at this stage, I think we're all very much looking forward to better understanding the new guidelines of WHO, as well as its implications for the protection of human health. So Dr. Reeder, the floor is yours. Thank you very much.
Thank you, and thanks to the organisers for inviting me along today to give a little bit of our perspective on this really important topic. I just wanted to start really by a little bit of a news around WHO. Of particular relevance to this is that as part of the transformation last year, for the first time, WHO formed a science division and actually created the position of a chief scientist to head this, Dr. Swaminathan. So for the first time, it's pulled together the major research entities, organizations like TDR and HRP, along with the quality assurance and norms and standards, so the actual guidelines from WHO, and then also created a new Department of Research for Health to try and give some strategic guidance across the organization. So this means not that this department has taken over research in WHO, but it gives us the opportunity to stretch across the organization and pull together opinion on particular issues. And one of the issues, of course, that was one of our priorities was how does WHO address the questions being asked by its member states about gene drive and genetically modified mosquitoes. We've taken three approaches to this, the first being technical, the second ethical, and the third being regulatory. But I want to start back where Claudia mentioned earlier, that this isn't so new for us. And in fact, in 2014, TDR published this guidance on testing of genetically modified mosquitoes. But the field has moved really quickly. In 2014, CRISPR was just, a really novel technique. This year, the Nobel Prize was given for it. By 2018, gene drive had become a real prospect to be addressed. So we need to update this. And one of the first things we've done is we've been reaching out to the many collaborators around the world who were a part of the initial framework and particularly updating so it includes guidance on gene drive as well. And this should be coming out early in 2021 as suggested. But already we're seeing the drafts and the themes, and we felt that WHO needed to move along with the work it's been done in ethics and regulatory to be very clear about its position on here. So just a couple of weeks ago, I launched this document, which is easily available on the WHO website, which is really stating in broad terms our position on genetically modified mosquitoes, including gene drive at this time. And while I encourage you to have a look at this, I can really reduce this to three things. One is we really do need help in tackling vector-borne diseases. And we're looking at deaths of above 700,000 people a year, but massive impact on economy, on well-being across the world. And as we're looking at it today in the context of COVID, we're likely to be seeing some of the gains that we've made in vector control sliding backwards a little bit as health systems are weakened by the current pandemic. So there's a really strong case for us getting new tools and really trying to address vector-borne diseases. The second, I think, is the most important to be clear about, is that WHO is really taking a position, and particularly with the new science division, of encouraging innovation, including things like genetically modified mosquitoes. And we really want to see a good evaluation of the potential of tools like this to contribute to the global fight against vector-borne diseases. So we're enthusiastic to look at innovation and look at new technology coming through. But, and I think this reflects the same position that most people have talked about, we need to do this in a really considered, sensible, safe way. And so we're talking about within this document the stepwise evaluation of making sure that we know where we're going before we get beyond the point of no return, making sure there's good evaluation done at many levels. So please do have a look at this, and I think it's pretty clear about where we're standing, and we're keen to see these technologies tested, but tested properly. One of the other things that rose when we were revamping the earlier guidance is we probably didn't spend as much time looking at particular ethics. And before I move on to that, please, I will give the email address that we are looking for comment on that position paper. So if you have things to say, please do get in touch with us, and this will be monitored. So at the same time as we launched this, and I think it was a really good to do these at the same time. It was the realization that the success of introducing new technologies like this is as much about its engagement with community and the issues that community have and the ethics around it, as it is about the technicalities that need to be solved in here. So I also would encourage you to go along to the side and have a look at the newly released guidance on ethics of vector-borne diseases at this stage. And again, I'm not going to go into any detail. We're limited for time here, but I think the take-home message of this is it's absolutely critical that we engage with communities in design and implementing of the testing of these technologies in order to really have a strong community base and ethical path forward in these issues. The third thing I wanted to mention was our role in regulation, and there are two aspects of this. One of the aspects is what do we do about looking at emerging technologies? And of course, Gene Drive is very much in that. And in 2012, WHO established the WHO Vector Control Advisory Group, VCAG, to start having a look at these issues, not just Gene Drive, but a whole range of different issues around vector control. And this has been a really successful process and given a really robust debate and information that's crossed the different disease programs within WHO. And currently, the VCAG has got a couple of different technologies under review at this stage, and is looking very carefully at these. But then we need to plan for the future. What happens when we start to see the potential of Gene Drive coming through, or other genetically modified organisms coming through? How do we make sure we get them out and get them into use? And so already, although it's quite early days, as we've heard earlier, the WHO pre-qualification and regulation department is starting to gear up about how they would approach, how they plan for success if it comes in this area. And a lot of this is making sure that beyond regulation, that this is monitored carefully, and the effectiveness of the products and the safety of the products in the long term is very well monitored. So I'll keep within the time here, but just to say that from our point of view, this is a technology with immense potential. We really want to move forward and make sure that potential is explored, but we are really giving guidance on both ethical aspects, technical aspects, and also gearing up internally about how we can have a smoother regulation of these programs. So we really hope to work with many of you, and I want to thank the many of you online today who've contributed to these documents in the past as well. So thanks very much, and I'll end it there.
Thank you very much, Dr. Reeder, for your intervention. A big thanks also to, on behalf of the Secretariat of the Intergroup, to all previous speakers of today's webinar. Unfortunately, due to unforeseen circumstances, Ms. Rodriguez Ramos will not be able to participate to today's discussions. However, she sends her best regards and kindest apologies with reference to this development. Therefore, moving ahead with our events agenda, with the kind help of technology, I will be moderating the Q&A session based on the questions received by the audience. Perhaps worth to underline at this stage that while we will try our best to answer all the questions received, I'd like to proactively state that there might be a need to proceed with a selection depending on time available, but also on the number of questions received. So the first question would be for Dr. Burt. Perhaps Dr. Alfie as well could provide us with a short comment. The question reads, we've heard about gene drive, but what's the difference within that and also gene editing? Specifically, why gene drive would be considered a useful approach for malaria or, for instance, for dengue? Mr. Burt, you have it.
So thanks for the question. So in terms of applications, or leaving aside pure science for the moment, gene editing is usually used to change the properties of organisms over which we have a fair bit of control. So like crops in a farmer's field or livestock or microbes that we're growing in a vat to make some particular chemical. And gene drive is when you want to have some control over a population which you don't normally don't have as much control. So a pest population, the reason it's causing problems is we can't control it, and so it's a new approach to finding ways to control that population. And it's got this property, the gene drive approach has the property of being where the gene transmission from one generation to the next is changed, it's non-Mendelian, and so that allows the gene to spread through the population when otherwise it wouldn't. And whereas for conventional gene editing, and I'll say there are many, the field of gene editing is much larger than the field of gene drive. Many more scientists working on it, and it typically does not have that property. Typically, the transmission of genes from one generation to the next is not changed with gene.
Thank you very much. Dr. Alfie, perhaps a follow-up comment from your side as well?
Sure. So absolutely, as Austin said, gene editing for me is a method of modifying DNA, which has a very wide range of applications in a very wide range of contexts. Whereas gene drive is a phenomenon of genes spreading in populations. Now, to make a artificial synthetic gene drive might use gene editing if you take a broader definition of what you think gene editing is, but that's really the connection. Gene drive could be made with gene editing. Gene editing has a very, very much wider range of applications.
Thank you very much. I see that through the platform, there is a second question as well for Dr. Alfie. Specifically, what are the main environmental risks of gene drive approaches that you are currently working
on? Well, for a proper risk analysis would cover a very, very wide range of possibilities, including lots of things that aren't very likely. So that, I'm not going to attempt to be comprehensive in this, but you can think of lots of different types of risk. There's one class of, what if it doesn't work? So it doesn't do what you think it's going to do, and you don't get the benefit that you intended. Then you've spent some money and done some things and not received much of a benefit. And from a developer's point of view, because these are actually quite hard things to develop, that is what we think about most of the time of getting it to work in the first place. And then there's another sort of class of risk. Most of these are not very unique to gene drives, but what if we succeed? What if we do what we intended and suppress a particular population or reduce transmission of a disease? Did that have some beneficial consequence that we weren't aware of that we've now perturbed or lost? So a pest population particularly, you might know that it does some bad things, but does it perhaps do some good things or some ecosystem effects that you weren't aware of? So these are quite general sort of things. There's another issue about, which I guess applies to a general concern about a lot of genetic technology, or will it get into things that you didn't intend it to get into? And there's actually another question about this, which relates to species specificity. These systems are very mating based, and so indeed species was a shorthand for species or species complex which can form fertile hybrids. Most of the designs are such that if they got into some other species by some other means, which is pretty hard to imagine, other than by mating, it would be extraordinarily hard for them to propagate. They tend to depend on very particular sequences or properties. So that the sort of horizontal gene transfer risk seems very low for most designs. All of these things need to be looked at on a case-by-case basis, of course. But those are sort of some of the categories of things people talk about, but in no means comprehensive sort of way. I would point to the sort of thing that Patrick was talking about of some of the risk assessments of ACCIA, for example, a natural and gene drive cover the kinds of range of risks people have considered.
Thank you very much for your input, Dr. Alfie. I also see, in an attempt to cluster some questions together, I also see, since we have addressed risks, there is also a question I understand most likely targeted for Dr. Alfie, but also Dr. Burt, that reads out, what benefits can be associated with the potential use of gene drive applications in Europe in the future? And could you please provide some concrete examples? That would be once again.
Sorry, could you just repeat the question, please?
So the question would be, what benefits could be associated with the potential use of gene drive applications in Europe in the future? And could you provide some concrete examples?
Right. So the two broad areas that benefits or purposes, objectives for developing these gene drives are for vector-borne diseases and for the control of invasive species in the context of conservation. So those are the two areas where there has been the most thought and the most research. As Luke mentioned, there's also some possibility and some thought going into whether some of these approaches might be relevant for agricultural purposes, but that's kind of less well developed than the other two. In terms of vector-borne diseases, the major ones, of course, that have been mentioned are malaria, dengue, Zika, etc., sleeping sickness, etc. And as Claudia mentioned, these are often diseases that attack the poorest and most vulnerable people in society.
So if I were to add to that, I think that if I heard correctly, part of the question was about a European context. So the remote regions of Europe. So Madeira, which is part of Portugal, since we had a few Portuguese affiliated comments earlier, has Aedes aegypti, which transmits dengue, and indeed dengue transmission, and so certainly to some French overseas territories, and I think also some others. So for the remote areas of Europe, definitely there are some obvious relevance just in terms of vector-borne disease. In terms of continental Europe, there is low level of transmission of dengue in France and Italy and so on, transmitted via another Aedes albopictus, which is also a major fighting nuisance. So clearly Europe is not the most affected, continental Europe is not the most affected area if you're talking about mosquito-borne disease, which is more tropical areas. Then I think you need to think of Europe's commitment to sustainable development goals and so on, as mentioned by Claudia, for which vector-borne, mosquito-borne disease is really important, and then also some of the conservation applications similarly.
Thank you very much. I see a couple of questions popping up also addressing Dr. Rudolfsheim intervention. So one would be, what should EU member states be doing, as well as prioritizing, to support responsible gene drive research in Europe, as well as with developing partners, such as countries in Africa?
Yes, thank you very much. I think it's clearly it's an interesting question in terms of the difference between what we see in Europe, what we see in other parts of the world, and we should not underestimate the how people look at Europe as a reference for some of these developments. I would say, in particular, from interesting and how Europe acts on some of these development points are. In terms of creating an environment, clearly Europe has an environment, and as was reported at the beginning, it's obviously strong on making sure that we protect the environment, that we understand what the impacts are. So if you look at what Europe can do in this respect, then I would say three aspects. First of all, it should be clear that Europe has very specific policy priorities. Yeah? And those policy priorities may be different. I would even suggest are very different when you're looking at countries, sub-Saharan Africa or Asian countries, where indeed some of these diseases, as was pointed out, create really big trouble, and we are in need of solutions. So the policy priorities, why Europe takes positions on certain points, in particular on synthetic biology or on GMOs, to clarify that, to understand that there are differences in policy priorities is an important communication point. The second thing about the fact that we do have, and we are proud about, stringent regulatory conditions, but they fit within the precautionary approach. And there's a lot of different interpretations on what a precautionary approach is. In many cases, people see this as an opportunity to stop research, which in fact is not what a precautionary approach is about. The Commission 2000 already had a wonderful communication on how to implement the precautionary approach. Unfortunately, too many people have forgotten about that wonderful paper, and we should probably take it up again to show people that it's not just about stopping, it's about creating a responsible environment. Finally, I would say I think Europe, 30 years of biotech-related GMO legislation, can play a role in bringing the lessons learned from those experiences to others, to share that, rather than having people to start and go through a similar process. Try to understand what are the real important points, how do we use our risk assessment in a way that indeed it serves that precautionary approach, and not just to put unworkable limits, but to show an environment in which you can proceed in a responsible way.
Thank you very much, Dr. Rudelsheim. I see also a very interesting question. Are current risk assessment guidelines keeping pace with technical developments? I think it would be interesting to receive your point of view on that.
I thought that was breaking up at one point.
I'm sorry, I can repeat. Are current risk assessment guidelines keeping pace with technical development?
Yeah. There are two aspects to that. I think one has to do with the framework in which you provide the kind of risk assessment. And I think a lot of people have been discussing whether can you have a general framework, whether that's developed through EFSA, for instance, or whether it's part of the CBD processes. I think the process of doing the risk assessment is still, as far as we see today, holds. And from our own experience, we find for doing risk assessments, where you see the gaps, of course, is that in fact, when you look at a specific technology or when you look at a specific species, you may lack information. Those are the parts where we need to have the information that feeds the risk assessment. So when we say you can use the existing systems, indeed, I think we have not seen that there's a major failure in any of the procedures that we're using, but where we, depending on the application, where indeed we need more information is maybe information on the environment, information on the species. But when you focus your research on that, you can fit it into the system to do your risk assessment.
Thank you very much, Dr. Rudelsheim. I see questions continue to pop up. I'll just try to perform my best time-wise to address as many as possible. There's a question for Camilla. Camilla, you mentioned the need to balance risks and benefits within your intervention. The question reads out, how are the existing guidance and regulatory framework addressing this need?
Okay, thank you. So it very much depends on the jurisdiction that the legislation is taking place in. But in Europe, the EU legislation focuses only on risk and does not, in its evaluation, take benefit into consideration. The benefit is taken into consideration at member state level, where risk managers and members within the member states make the decision if there is a benefit to be had to introduce these technologies. In other countries, it's different, where both can be combined, particularly in Africa, how they've interpreted some of their legislation.
Thank you very much. Moving ahead, there is a question for Dr. Claudia Emerson. So there seem to be concerns that if your organizations could own gene drive technologies once finally developed, do you think this is a potential risk?
Sorry, Elias, could you repeat the question? I didn't catch that first half.
Yes. So there seem to be concerns that if your organizations could own gene drive technologies once finally developed, do you think that this is a potential risk?
Sorry, it's the audio. So I'll just repeat it back and see if I've got that right. So there seems to be the impression that there are certain organizations that don't think that gene drive technology will work.
That a few organizations could own gene drive technologies
Oh,
once
I see.
finally developed, and if it poses a potential risk.
Yeah, so this is a question that's come up about the privatization. So right now, it depends on how we want to, how we understand the kind of risk that it introduces. So certainly the current context and what's underway right now is that the application, certainly from the public health aspect, which is the one that I'm most familiar with, is that it's being done for the public good. So there aren't profit-making motives involved in this. The technology is being co-developed with partners, and I believe Dr. Bird is probably in the best position to actually respond to this. So I think from an ethics perspective, the idea is that these are technologies, if we're going to use gene drive technology to advance public goods, then we should stay focused on what those goods are and try to manage, of course, any kind of attempts to privatize this and in a way that would deprive equitable access and see through the full complete benefits of the potentials for technology. So it certainly is a risk, but I don't think it's a significant risk at the moment, given where we are in the field.
Thank you very much. Moving on, there is a question as well for Mr. Kent Redford, chair of IUCN Task Force on Synthetic Biology. So within your intervention, one of the key messages of the technical assessment by IUCN is that conservation action needs new tools. Could you please provide us with some concrete examples also beyond gene drive? Why is it specifically important for IUCN to look at the issue of symbio?
Yes, thank you. I appreciate the question. The answer to why new tools are needed is really contained in the latest publication by UNEP, which showed that the conservation goals have not been met globally, and in fact, we've moved backwards in some of the cases. So it's clear that the traditional tools available to us, the way we've been deploying them, has not worked. And the answer, we believe, is not that you throw those tools away and find brand new ones, but rather that you work with existing ones and then seek to see whether there are other kinds of tools which may complement those and increase the efficacy and allow us to better try to approach these global goals. So it's in that context that the synthetic biology question set of tools is being asked. In terms of the question, the portion of the question regarding new tools, I just run through 3 or 4 of them. Environmental DNA is proving to be looking as if it's a very effective way of doing monitoring and detection, particularly of rarer species. Most interestingly, as far as I'm concerned, is people catch leeches and see what blood meals they've fed on, and then determine the presence of very rare species by looking at the leeches themselves. But it's also being used with dung beetles and mosquitoes and a variety of things. So that's one. The second one would be audio monitoring, using advanced intelligence, AI work, artificial intelligence, as a way of taking very, very long time periods of acoustical recordings in particularly rainforest areas so far, and then using these AI tools to determine changes in community structure based on different kinds of impacts, for example, with selective logging. And then third, the use of drones, which has really revolutionized all sorts of different sampling. I just say particular the presence of sea turtles close to nesting beaches, which has never been we've never been able to really get a good handle on it. And drones are providing outstanding new data sets for assessing conservation effectiveness. Thank you.
Thank you very much, Mr. Redford. I also see a couple of questions for Dr. John Reeder as well. So to begin with, how does WHO's role in providing guidance for gene drive research complement the work that is done under CBD?
I think that's a relatively easy one to answer, because we're very much paying attention to the other conventions and what's going on around. I guess what we are trying to determine is not so much making a judgment in that kind of policy level, but how do we condense that to answer the questions our countries are asking them when they're faced with the prospect of possibly testing some of these techniques in the end. So I think, without being long-winded, very simply, we are very carefully looking at this and trying to fit our guidance within the bigger policy frameworks. And in some ways, we're taking very much a very practical view of how we condense the many different pieces of information. Because I think one of the advantages or maybe disadvantages of being in WHO is that people come to ask us for this advice on health issues. They will not go and look at the entire spectrum of activity around this. So we're trying to condense this and go along with that.
I see that perhaps time would allow for a second follow-up question, once again for Dr. Reeder. If a country was interested to use a gene drive tool for malaria or dengue control in a few years, how would specifically WHO support that country?
One of the issues here is we are, of course, trying to let countries make their own decision about this. But as with any country, if there is a need for assistance with capacity to do this, or in terms of trying to look to the guidance framework, we'll be looking like with any disease. I mean, if you're now looking about advice on insecticide-treated bed nets, there are technical people from WHO who will come and advise and will come and assist with this, and we hope it will be a similar kind of picture into here. But I guess what we are not in the business of doing is doing the trials within country. We want to make sure that the questions countries need to ask are very clearly articulated within the guidance, then they can make truly informed decisions around this. But as with any other health issue, I mean, people can approach the WHO for assistance with these things, and we give what assistance we possibly can.
Thank you very much, Dr. Reeder. I see that timing would allow us perhaps for a final question to Dr. Burt. Dr. Burt, the question reads out, what about the risk of unintentional spread to non-target species?
Thanks for that. So there's two ways I think one can imagine, or the field conceptualizes unintentional spread. One has been mentioned already by hybridization. So to a different species. So as Luke mentioned, they can transfer by mating, and if those hybridization leads to fertile offspring, then potentially it could spread to another species. So that's something that very definitely needs to be considered and assessed before any kind of release. Are there other species that your target species could mate with? And do they make fertile progeny? And if the gene drive got across, would it work in that other species? And if it did and spread through that other species, would that lead to harm? You know, so all these things are very individual, case-specific questions that absolutely need to be addressed before you would do any kind of release. There's another kind of something that happens much, much, much more rarely, of horizontal gene transfer from one organism to another, not through mating. It's kind of through, it's kind of unknown still how that happens, but that's something that really only happens on an evolutionary timescale of millions of years. And so as all the sequencing data, these data sets are getting larger and larger all the time, that doesn't seem like that's a risk for most of the cases that have been thought of or have been thought about on the sort of ecological time frame that all of us are thinking. But again, that's something that needs to be addressed on a specific basis to make sure that general impression is going to be appropriate in the individual circumstance. I suppose I could also add, there is potential of transmission not to a different species, but also just to a different population. So if you want to control an invasive species, you want to control it where it's invasive and not back in its native range. And that's actually a very active field of research by geneticists trying to figure out different ways that you would restrict your interventions just to where you want it to be restricted. And there's half a dozen ways of doing that, and all still needing to be assessed, but people are on the case.
Thank you very much, Dr. Burt. Big thanks also to all of our speakers for today's webinar. Thank you very much also for your answers with regards to this Q&A session. At this stage, without further ado, I would like to provide the floor to MEP Maria de Grasa Carvalho for her closing remarks. Thank you very much.
Thank you very much. First of all, I would like to thank everyone involved, the moderator, all the speakers, and everyone involved also in the Q&A session, and that have contributed to a very lively and fruitful debate. Today's discussion have shown the potential benefits that gene-drive research could yield. They have also demonstrated that there are concerted efforts to ensure that research is carried out responsibly. It's clear that rules under the Convention on Biological Diversity apply to gene-drive research like to other modified organisms. At national level in the EU and elsewhere, national authorities are the ones to authorize and set the terms for gene-drive research taking place in their territory. It is therefore important to support information sharing about this research so decision makers are equipped to understand and make decisions based on sound evidence. Gene-drive research remains at the early stage and many questions cannot yet be answered, but hopefully as research progresses, the benefits, the risks of different use of this technology will become clear and clear. Consequently, further research can inform national decision-making on whether to allow or not different use of the technology. On the other hand, and despite progress made in the recent years, any release of gene-drive organisms for research purpose seems to remain several years away. As a global leader on biodiversity and public health policy, this topic is particularly important for the EU. Understanding the needs and perspective of EU researchers in these fields would be valuable. Final, and this would be my last point, the importance of gene-drive research is not limited to Europe, but is also very relevant to our partners in other regions of the world, notably African countries that have expressed interest in this research for the purpose of malaria control. In closing, ladies and gentlemen, I wish to thank once again to have joined us today and to thank the Secretariat of the Intergroup for having organized this very interesting webinar. Thanks a lot.
So on behalf of the European Parliament Intergroup Secretariat as well, I'd also like to thank once again hosting MEP Ms. Carvalho, but also big thanks to all of our speakers and participants for today's webinar. It's been a real pleasure having this event with you, this online event with you today. So very much looking forward to the ones to follow. Until then, all the best. Once again, thank you very much and stay safe. Thank you.